I'm Lisa, I'm a PhD student at the Department of Zoology, and today I'm here with Eva.
Hi, I'm Eva Higginbotham and I'm a third year PhD student at the Department of Zoology, and I'm a part of the Neural Network group.
Great, so could you tell us what are you studying?
So my PhD is about how we build a brain. So building a brain is an incredibly complicated task, as you might expect, and partly that's because brains are made up of lots of different components that all have to work together in order for your brain to function. So the most important of those components are called nerve cells or neurons, and these form connections with each other and pass on messages, and that's how your brain and your nervous system actually works. And so it's really important that you have the correct type of neuron at the correct place at the correct time, and there are lots of different kinds of neurons that you can be. And so my PhD is trying to figure out, how does a neuron know what kind of neuron it's supposed to be? And the work that I do is actually in Drosophila melanogaster, which are fruit flies, instead of in people. And partly that's because fruit flies reproduce very, very rapidly, and we've been using them for scientific experiments for over 100 years. So we actually have a huge amount of knowledge to draw on when we do experiments. And really importantly, although they look very, very different from people, there's actually a huge amount of similarities. So not only do we share something like 60% of our DNA with fruit flies, but actually their brains communicate in the same ways that ours do. So in both flies and humans, you have these neurons that are making connections with each other, and the way they pass on information is through neurotransmitters. So neurotransmitters are these special molecules that a neuron makes in order to communicate. And there are different kinds of neurotransmitters. And what kind of neurotransmitter a neuron really defines what kind of neuron it is. So my project is trying to answer the question, how does a neuron know what kind of neurotransmitter it needs to make?
That's really interesting, but also sounds really complex. So how do you actually study those things?
So I actually work in the Drosophila larva. So Drosophila lay fertilized eggs, and after a period of 10 days they hatch out as adults, but in that process a lot of changes take place. So the stage that I'm looking at is the Drosophila first instar larva, so that's the first stage after they've hatched out of the egg, and that's because their nervous system at that point is actually much, much simpler than as an adult, but it does have a fully functioning nervous system. So it can move, it can eat, it can respond to certain stimuli. And so what I do is I take the first instar larva and I dissect out its nerve cord and its brain lobes. So that's essentially the combination of, like with a human you have your brain and your spinal cord, so it's their equivalent to a brain and a spinal cord, but they don't have a spine because they're invertebrates. So I dissect that out and I stain it with different chemicals, different antibodies, and then I look at that under a microscope. That's basically what I do. So we have most of the images that I take of my nerve cords are at 63 times magnification. So it's a combination of sort of dissection skills as well as sort of chemical staining skills, and then using a microscope.
Yeah, so it sounds like you really love developmental biology.
Yes, I really like it, and I think I was very fortunate to be at a university like Manchester for my undergrad, which allowed me to study lots of different things within biology, because I discovered I love development, and Cambridge runs a 4-year PhD program in developmental biology, and that's the program that I'm currently studying on. So in this kind of program you spend your first year doing rotations where you work in different labs in different departments, and you just sort of try out different things and see what suits you best. So I worked with fish, I worked with flies, and I worked with stem cells. And when I came to the Department of Zoology in the Landgraf lab, where I am now working on flies, I knew pretty early on that this was the place I wanted to be. And that's partly because the project really suited me. Like I said, I'm looking at neurotransmitter specification, so how does the cell decide what kind of cell it's going to be, and that's really the core of developmental biology and why I like it so much. But also the department is super, super friendly. There's a lot of friendships between people in different groups and people who work different jobs here, and it's an incredibly diverse department. So I'm a fly person, you're a bird person, there are burying beetle people, and people who work on all sorts of things, and that makes it a really exciting research community to be a part of, and it certainly makes sort of research seminar days within the department really, really interesting, because you kind of never know what you're going to learn before you walk in.
Yeah, definitely. How about outside the department, how has your experience in Cambridge been so far?
So I really enjoyed being at Cambridge, and part of that is because of the college system. So I'm at Newnham, which is one of the women's colleges, and since I've been at Newnham, I've been really involved in college life by being a part of the MCR committee, which essentially is the group of students who organise events for the graduate community at a college and also are sort of represent the graduate community. So in my first year I was one of the women's officers, and we ran documentary screenings and panel discussions talking about all sorts of things, from sort of the women's role in ending the civil war in Liberia to the ethics of egg freezing, and there was some evolutionary psychology in there and that sort of stuff, so that's very interesting. And then later I became the president of the committee, which was very challenging because it's an 18-person committee and you're running events and representing hundreds of students. And also acting as the first point of contact for people outside of the college. So it's very challenging on top of doing PhD work, but I did really enjoy it, and I learned a huge amount, so it was partly just organisation skills and also communicating with lots of different people, because sometimes I was working with the principal of the college on sort of policies within the college, and sometimes I'm talking to undergrads, and sometimes I'm running parties, and sometimes I'm running serious things. So it was very hard, but it was really enjoyable, and I got a lot out of the whole experience being very involved in college. So that's one of the things I feel lucky about being at a university like Cambridge, which has the college system, because I think it's really good to force PhD students, to get to know each other in colleges like that.
That's definitely very important. Thank you for your time, Eva, and best of luck for the last year of your PhD.
Thank you.